Genomics is an interdisciplinary focusing on the structure, function, evolution, mapping, and editing of genomes. A genome is an organism's complete set of DNA, including all of its genes as well as its hierarchical, three-dimensional structural configuration. In contrast to genetics, which refers to the study of individual genes and their roles in inheritance, genomics aims at the collective characterization and quantification of all of an organism's genes, their interrelations and influence on the organism. 4. Genes may direct the production of proteins with the assistance of enzymes and messenger molecules. In turn, proteins make up body structures such as organs and tissues as well as control chemical reactions and carry signals between cells. Genomics also involves the sequencing and analysis of genomes through uses of high-throughput DNA sequencing and bioinformatics to assemble and analyze the function and structure of entire genomes. Advances in genomics have triggered a revolution in discovery-based research in systems biology to facilitate understanding of even the most complex biological systems such as the brain. The field also includes studies of intergenomic, within the genome, phenomena such as epistasis, effect of one gene on another, pleiotropy, one gene affecting more than one trait, heterosis, hybrid vigor, and other interactions between loci and alleles within the genome. From the Greek gamma epsilon nu gen, gene, gamma, epsilon, nu, epsilon, meaning become, create, creation, birth, and subsequent variants, genealogy, genesis, genetics, genic, genomere, genotype, genus etc. While the word genome, from the German Genom, attributed to Hans Winkler, was in use in English as early as 1926, the term genomics was coined by Tom Roderick, a geneticist at the Jackson Laboratory, Bar Harbor, Maine, over beer at a meeting held in Maryland on the mapping of the human genome in 1986. Following Rosalind Franklin's confirmation of the helical structure of DNA, James D. Watson and Francis Crick's publication of the structure of DNA in 1953 and Fred Sanger's publication of the amino acid sequence of insulin in 1955, nucleic acid sequencing became a major target of early molecular biologists. In 1964, Robert W. Hawley and colleagues published the first nucleic acid sequence ever determined, the ribonucleotide sequence of alanine transfer RNA. Extending this work, Marshall Nirenberg and Philip Letter revealed the triplet nature of the genetic code and were able to determine the sequences of 54 out of 64 codons in their experiments. In 1972, Walter Feers and his team at the Laboratory of Molecular Biology of the University of Ghent, Belgium, were the first to determine the sequence of a gene, the gene for bacteriophage MS2 coprotein. Feers' group expanded on their MS2 coprotein work, determining the complete nucleotide sequence of bacteriophage MS2 RNA and simian virus 40 in 1976 and 1978, respectively.